Now we will discuss stratified epithelial tissue. Last time we talked about simple epithelia, which if you remember was a single layer of epithelial cells. Stratified means that the cells are multi-layered. There are four stratified epithelial tissues that we will discuss today. Remember that this is the basement membrane that anchors the tissue, which you can see has been highlighted in each of the four tissues. You can now see that we have many layers as opposed to the single layer we saw in the last lab. Each of the four tissues is classified by the shape of its surface cells. You can see that the highlighted surface cells are flat as opposed to some of the other tissues that have surface cells that are square or rounded. The first tissue on the very left is stratified squamous epithelium, keratinized. It is stratified because it has multiple layers, and it is squamous because its surface cells are flat. Notice that the surface cells have no nuclei, which means they are dead cells. There is also a layer of keratin, which is why it has its name. The next tissue is stratified squamous epithelium non-keratinized. Notice that it does not have the surface layer of dead keratinized cells, which is why it is known as non-keratinized. These two are really considered the same tissue, but it just has two different varieties, so we will treat them each individually. Next is stratified cuboidal epithelium. It is usually two or more layers of squarish, round cells. And last is transitional epithelium. It is called transitional because it can stretch, therefore its cells go from a round shape when relaxed to a flat shape when stretched. Lastly, there technically is a stratified columnar epithelium. However, your textbook says that it is too rarely found in the body to be worth our consideration. Keratinized stratified squamous epithelium is found in the epidermis, which is the outer layer of skin. It consists of multiple layers of cells that becomes increasingly flat and scaly toward the surface. The surface is covered with a layer of compact dead cells without any nuclei. The cells on the basal layer, which is the bottom layer attached to the basement membrane, are usually round. The cells from this bottom layer are constantly dividing to produce new cells that push older cells to the surface until they eventually die and flake off. This is an example of thin skin. Over here to the left you can see a hair follicle. Let's move in closer to see more details. This top, darker stained layer is the epidermis. Below it is the dermis, which is made of connective tissue. The wavy boundary between the two is the basement membrane. Let's go ahead and zoom in on the epidermis. Now we can see the keratin layer. Because skin is exposed to air, it is keratinized to protect the surface from abrasion and is lubricated by glycolipids to avoid drying out. This is an example of thin skin. We know this is thin skin because the keratin layer is thin and has less layers of stratified squamous epithelial cells. If it were thick skin, the keratin layer would appear much thicker, as well as the epidermal layer. Here is a picture of thick skin for comparison. Notice how thick that keratin layer is. Thick skin is found most notably in the palms of the hand and the soles of the feet. Next, we are going to look at non-keratinized stratified squamous epithelium. It has the same description as the keratinized variety, except that it lacks the dead layer of keratinized surface cells. This example comes from a small slice of an epiglottis. The entire anterior surface, which is the part of the epiglottis facing the tongue, and the posterior surface, which is the part facing the larynx, are covered by a stratified squamous epithelium. These areas are vulnerable to abrasion due to the passage of food. This is a real good example of non-keratinized stratified squamous epithelium. Notice the cells on the bottom layer would be attached to the basement membrane. The pink staining connective tissue is on the opposite side of the basement membrane. The basal cells are more round, and as we move up to the surface cells, we see that they become flattened. The best way to differentiate this from its keratinized variety is that there is no wavy layer of dead keratinized cells at the surface. Keratin is not necessary in this tissue because the epithelium is not exposed to the drying effects of air, and it is constantly kept moist. Next is the stratified cuboidal epithelium. It is composed of two or more layers of cells. The surface cells are square or round. Stratified cuboidal epithelium has a limited distribution. It is most often found in large ducts of exocrine glands. Typically, it has only two layers of cuboidal cells. This image is of an esophagus, which has a few examples of some ducts lined with stratified cuboidal epithelium. Here is an example of a duct with 2 to 3 layers of cuboidal-shaped cells. The purple dots are the nuclei of each cell. The large opening is the lumen. Last of our stratified epithelial tissues is transitional tissue. This tissue is specialized for stretching. When the tissue is relaxed, it somewhat resembles a stratified squamous epithelium, but the surface cells are rounded, not flattened, and often bulge at the surface. It is typically 5 or 6 cells thick when relaxed and 2 or 3 cells thick when stretched. Some cells of this tissue even have 2 nuclei. Transitional epithelium is also called urothelium because it is a specialized stratified epithelium found in the lower urinary tract. It rapidly adapts to distension and contraction by changing from a taller to thinner epithelium. This tissue section is an example of a ureter, which is lined with the transitional epithelium. From this view, this tissue kind of looks like a sand dollar. When we zoom in, you can see several layers of cells and also the large dome-shaped umbrella cells on its surface. This tissue would look much different if it were stretched, but since we don't have an example to show you, then we won't worry about it.